thank you, Pascal. So thanks a lot for the invitation. It feels great to be here. It feels even better after this COVID period, which is probably not over, but having been stuck at home for such a long time, so happy to be here, especially since it's where all started when I was a student with actually Pascal almost 20 years ago. So just like you, attending seminars and classes and so on. So I feel particularly happy to be here now in front of you, trying to tell you about what we are doing. So today, so what I originally planned was to actually tell you, I thought it was a seminar. So I thought, yeah, okay, let's take the last study that we've done, and I'm going to go into the details and so on. And then I saw that it was more intended to be a course or a lecture, and I thought that instead of going into the very details of a small aspect of what we're doing, I wanted to provide you with a broader perspective on the kind of interest that we have and the methods that we develop and so on. So this is what I'm going to do today. Like it was said before, feel free to interrupt. I'm very happy to take questions. My goal today is to convey a message, and so if I need to answer questions, even basic questions, I'd be very happy to do so, because that's the only thing that matters to me. Okay, so we have various interests related to the control of growth and proliferation using budding yeast as a model system. So this is a budding yeast here that you can see that basically generates a bud, and this bud grows, and ultimately you have a nuclear division that leads to the formation of a mother and a daughter cell. So it's a very specific division pattern because it's asymmetrical. It's been used extensively as a model to try to understand various aspects of control of the division process. One thing that we were sort of interested in is how actually cells as a species manage to control the broadness of the size distribution. Okay, so how do cells manage to control their size? This is one thing that is quite very theoretical questions, but in the end, people are trying to understand what are the underlying mechanisms and so on. So I will try to explain you what is the problem and how we can actually try to address it, or at least how we can make measurements that help address it. So that's one thing. The second thing is that obviously cells are constantly sensing and responding to environmental challenges, and so one of the examples I would like to provide today is how cells actually enter into this particular state that is called quiescence, during which they reversibly arrest their cell division cycle. And the last thing that is quite somehow related to the previous one is whether cells manage to undergo divisions in an unlimited manner, or whether they actually go through a state that is called senescence, which ultimately leads to cell death. And so what is the underlying mechanisms, and what is this that we are looking at when we look at all cells, dead cells, and so on and so forth. So these are mostly the three kind of questions which you see are all related, in the sense that for all of them, we're focusing at the division process, why it stops, why it does not, how it's controlled, and so on and so forth. So I won't necessarily go into all the mechanistic details, but I just want to raise the questions and also show you how the implementation of an experimental strategy can be helpful to actually put us on the way to solving these questions. Okay, so this is pretty much what we will try to do. So I will try to define the questions as precisely as possible, provide some insights about the methodology that we develop, the results that we obtain, without necessarily going into all the details. But feel free to interrupt. I'm happy to provide more details if you want. Okay, so there is this very long-standing question, which is how do cells control their size? So if you look at the distribution of cell size, okay, this is what you get here. I guess this is the pointer. Okay, you never get to have like extreme, cell size, even though you still have this extended tail and so on. And so the question that you may ask is, how do actually cells control their size? Okay, so the way to actually formalize this problem is very simple. If you imagine that cells are actually bioreactors that exponentially increase their mass over time, you can define, you know, this growth thing with two parameters. One is the growth rate, so the log 2 over the mass doubling time, okay, and the other one is the duration of the division. Okay, so if cells wants to achieve a doubling during the duration of the division, what they need to do is that they probably need to coordinate their growth rate with the duration of the division. Indeed, if the duration of the division is smaller than the mass doubling times, cells will shrink over time. I hope you had time. Oh, sorry, it's going too fast. So, just I need to learn how to use this thing in a. Okay. Somehow I'm the only one who has problems with that. But so yeah, so I was saying that if the duration of the division is smaller than the mass doubling time, then the cells would shrink over time. And on the other hand, if the duration of the division is longer, then the cells will increase their size over time. So basically, there is this sweet spot where you have a perfect coordination between the duration of the division and the growth rate, which is the only situation which can ensure a stability of the volume at division. However, like always in biology, there is noise, and noise is going to shift or make deviation, drifts in the distribution of cell size at division. And if you iterate this over time, you will actually see that the cell size over time, if you follow a single cell, it will basically diffuse away from its original size. So even if you take this condition, okay, you will never be able to obtain a stable distribution of size over time or in the population. Okay, so basically what we're saying here is that with this thing, this control mechanism in which you would have a perfect control on the timing of the division, you cannot ensure a cell size homeostasis. Okay, and there has to be therefore a coupling between the growth process and the division process. And a very easy way to implement this is to actually say, okay, look, when the cell reaches a given size, which we could refer to as VC, a critical size, then the division can occur. And obviously, so it's a very trivial way to implement size control, but if you implement this, then you're safe and you can have a stable size at division and ensure the stability of the size population over time and across the population. Okay, so that's fine, but that's very theoretical. So how do I actually address whether there exist such kind of mechanisms of control of cell size, sensing of cell size, and feedback between the cell cycle and the growth process? Okay, well, there is a way to actually do it, which is to, yeah, so I guess it's the old version of my presentation. Okay, no worries. There is a way to do it, which is basically to experimentally measure how much a cell grows during a cell cycle. So again, if you take this growth model, which is this exponential growth model, and if you calculate the variation of volume from birth to division, okay, in the case of a sizer, so I forgot to mention the name, but in the case you have a strong, you have a critical volume that needs to be reached in order to go through division, so this is what we call a sizer. This thing is fixed, and therefore there has to be a relationship between the variation of the volume and the volume at birth, which is such that you would get a slope of -1, as you expect, if this VDIV is indeed fixed. So this means that if for some reason a cell is born bigger, then it would have a smaller increase of cell mass during its division and the other way around. Okay, so if experimentally you're able to measure V birth and delta V, then you would be able to see whether there is indeed a size control mechanism occurring. Okay, so this is what we actually refer to as compensatory growth, just because what I just explained, that if you're small, then you need to grow bigger and so on and so forth. Okay. On the other hand, if the cell is actually behaving like a timer, then if you do the little math here, what you see is that there is actually a positive slope for this relationship between delta V and V birth. Okay? So this is good because this gives me an opportunity to see whether there is any mechanisms of compensatory growth during the cell cycle. And this actually has been done a long time ago. Actually, that's when I was born, so yeah, quite a long time ago. Working with S-Pombi, okay, as a model system. So Pombi is a bit different than budding yeast because it divides by fission, okay? And so what people have done at that time was to actually measure this variation of cell length. So it's an elongated cell, so it's very easy to measure cell size just by measuring cell length. So if you measure the variation of cell length as a function of the length at birth, you see that indeed there is this negative relationships between these two quantities, okay? Which indicates again that bigger cells, they actually grow less than smaller cells at birth, okay? So there is indeed such compensatory growth occurring, and it's actually, in Pombi, it's a G2 thing, so it's because these compensatory mechanisms occur during the G2 period of the cell cycle, ensuring that cells will enter mitosis only when they have reached an appropriate size. The same thing has been observed in budding yeast, so here it's not the original paper, but it's an interesting one in any case. So in yeast it's a bit different because it's the duration of the G1 phase and also the variation of volume during the G1 phase that is negatively correlated with cell size at birth. Okay. So in budding yeast there is a compensatory growth that occurs during G1, and obviously then the key question is that because it's G2 in Pombi and G1 in Cerevisiae, and because you could imagine that mechanisms are actually conserved between even distantly related species because cell cycle is heavily conserved, one may ask the questions whether there are also other mechanisms of compensatory growth that occur outside of G1 in budding yeast. So this is one of the specific questions that we got interested in and that I'm going to describe right now. The second question is, okay, you can see that there is relationships between variation of cell mass and initial size, so that's what I call compensatory growth. But how does this actually affect the overall size homeostasis in the population? So that's the second question that I was planning to tell you about. Okay, so for this, so what we do is that we use a simple microfluidic device in which we can put some cells, and cells are grown by diffusion. Okay, so you have like basically channels to actually replenish the medium, and therefore we can watch the growth of individual 2D colonies, as you can see here. Then we use standard, image processing techniques to actually get the contours and track the objects over time. So that's not a big deal, very classical. But then what we want to do is, because we want to identify new relationships between sizes, timings, and so on and so forth, we needed to have a clean way to actually measure clear points during the cell cycle so that we could measure the volume of the cells during S phase, during G2, etc., etc. So how do we do that? So for this we use actually a histone marker, and because histones are supposed to be synthesized only during the S phase, okay, you should expect to have a plateau, then a linear increase, then a plateau during G2, and a decrease in the signal upon anaphase of the cells. So indeed, this is what we get. So if this is a single cell that we track over time, and you see that we can easily fit a piecewise linear model to actually identify the different phases of the cell cycle. And based on this, so obviously we can do it on many cells. So this is just an example. So this is just 25 random cells. So we can sort of standardize the analysis and so on, so that we can compare all the data and try to identify these relationships. So altogether, we measure 34 variables, okay, which are like the volume at division or birth, well, same thing, the volume at the end of G1, etc., or the timing, the duration of the S phase, the volume of the bud, and so on and so forth. And we ask, is there any interesting relationships between all these variables? Okay, and this is what you get. So you can you can represent the heat map of correlation coefficients between all the variables. And one thing that you already find already is that you can recover, relationships that were already identified. So, for instance, here you have a strong negative correlations between the duration of G1 and V birth. So that just means that if the cell is born too small, then it has an extended G1 to in order for the cell to grow, and conversely, if the cell is large, then it just requires a short G1 before entering to S phase. So this is reassuring because it means that we can actually recapitulate results that were already known. Okay, so that's actually another way to plot it is to plot the variation of cell size during G1 as a function of V birth. And if you bin the data, you see that there is this negative correlation which recapitulates the known G1 size control thing. But now what is quite striking is that when we plotted this diagram, we also observed that there was actually some negative correlations here and there that were unknown. Okay? And this basically tells you here that there is a negative correlations between the volume of the bud at the end of the S phase and the duration of the subsequent G2M phase. Okay, so that you can plot same thing like this, and that basically tells you that there seems to be actually a size control mechanism that this time operates on the bud. Okay, which means that if the bud is too small by the end of the S phase, then the duration of the G2M phase has to be extended. Okay. So that's nice, but what does this tell me about the actual control of cell size? Okay, I have to say that there has been many studies that have attempted or even succeeded at identifying compensatory mechanisms between different, usually between the duration of G1 and the cell size and so on, but none of them are actually trying to see how the size distribution is controlled over time during a cell cycle. So here what you can see is actually the coefficient of variation for cell size as a function of cell cycle progression. Okay. And what you see already is that, so this is represented as a log-log plot, and this is a line fit. Okay, so that shows you one of the square root dependency as a function of the mean cell size. Okay, and that's a very expected thing. If you model growth as a sum of elementary growth processes, what you expect is that the CV should actually decrease as 1 over the square root of the mean cell volume, just because you're dealing with bigger and bigger compartments, and so you average noise out from the distribution. Okay, so this is pretty much what you get here, and that's just a statistical effect, right? But now, if instead of plotting the CV, you actually plot the Fano factor, which is basically the variance divided by the mean, then you get something that we can refer to as the noise strength, and that actually reflects the intrinsic variability in the distribution. And what you see here when you plot this noise strength is that it's not a flat curve. Okay, see that the error bars are pretty small, and there indeed seem to be a decrease in the noise between the beginning and the end of the G2M phase. And so indeed what we found is that not only we have a compensatory growth effect in G2M phase, but it's also concomitant with a decrease in cell size variability during that particular phase. Okay, and that, I mean, I'm not going to show the details, but that we found was actually very much dependent on the genetic background. So we have played with mutants in which we can perturb the compensatory effect, and this was actually also affecting the reduction of noise in the distribution. And therefore, we are sort of, quite confident that the two things, so the compensatory growth and the size homeostasis, the actual size homeostasis, are somehow related quantities. Okay. So the take-home message here is that in this study we have developed a single cell division tracking technique, so which allows us to monitor cell divisions with subcell cycle resolution. And that allowed us to actually identify a new mechanism of size control, which lead to the conclusion that it's not that there is only one phase of the cell cycle during which growth and division are coupled, but it's very likely that there are actually several phases during which this happens. So that's somehow the take-home message. The underlying mechanism, obviously, you know, we could ask, you know, what is the underlying molecular mechanism that actually drives this coupling between growth and division? And this is, I'm not going to go into the details, this is under strong debate, and there has been a huge contributions in the last 20 years, so if you want to know more, I'd be happy to tell you afterwards. Okay, so all of this was based on experiments in which we monitor the growth of the cells as here, and as I said, so cells grow exponentially, and that's nice, but if you look in the wild in particular, or even if you're in your test tube, when cells grow, they actually consume resources, and sooner or later they're going to deplete the medium from the nutrients. And it is very well known that cells, when they are actually deprived of nutrients, they would enter this so-called G0 state. And this G0 state is very interesting because it provides them with very interesting properties such as long-term survival and stress resistance. What is even more important about this G0 state is that it's a reversible state. Okay? So this state, we call it quiescence. Okay? It is very relevant in many biological contexts, and not only in unicellulars, because it is also known in the field of stem cells, cancer cells, etc., etc. And I have to say that it's a pity that most of the studies, you know, focusing on many different aspects of biology are actually using proliferative cells, in a proliferative state as a model, because in most of the contexts, if you look at the occurrence of this quiescent state in the wild, it's actually much more frequent than the proliferative state. Okay. So in the end, we know much more about what is going here than what is going there. Okay. And this is something that we thought, you know, could be interesting. And so before to study more carefully using single cell methods, and before telling you a bit more about that, I would just want to explain you a bit more what are these quiescent cells. So first, quiescent cells or starved cells display a massive reprogramming of their transcriptome. Okay, so this is just for illustrative, but you have many genes that are turned off. You know, the overall metabolic status is very different from that of the proliferative cells, but there are also genes that are turned on, and that, for instance, ensure stress resistance and that kind of thing. So massive transcriptional reprogramming, but also a huge change and a huge remodeling at the subcellular level. Okay? So this is one example. For instance, the mitochondrial network is greatly modified upon starvation. Okay? And it's also known that many proteins that are cytosolic proteins, this is an enzyme that is involved in an amino acid biosynthetic pathway, and that seems to form foci upon starvation. So there are, and there are dozens of examples like this. Okay, so basically, quiescent cells, they undergo this massive change in their subcellular structure, to the extent that even biophysical properties of the cells are modified. So it has been proposed that actually starved cells would undergo this glassy-like transition of the cytoplasm, which could have important consequences, which we don't know actually. So then, you know, a problem with quiescent cells is that when we refer to quiescent cells, we don't really know what we're talking about, because they are intrinsically more heterogeneous than the proliferative cells. So this is something that is observed, but the origin of this is not known. So this is one of the key questions that we wanted to address, developing a particular methodology to look at the cells as they enter into quiescence. So origin of heterogeneity is a big question, and it also, you know, the fact that there is heterogeneity is also the reason in quiescent cells is also the reason why people actually work with proliferative cells, just because they're more homogeneous and it makes their life easier. Okay. The second point that I think is very important is that people have looked into the response of cells to starvation by inducing an abrupt starvation like this. Okay. Okay, so you take your cells, they're happy, working glucose, and then you abruptly switch them to zero carbon Swiss medium, for instance. Okay, so this is good. This is easy. It's easy to proceed that way. But the point that I want to make is that if you look at an unperturbed life cycle, for instance, if you put cells in flask and you let them grow over time, you know, what you will see is that basically the cells are constantly modifying their environment, which in turn will affect how the cells are behaving, okay? How the cells respond. Okay? So you have this mutual, you know, interaction between the cells and the environment that is quite complex, can become quite complex. And actually, if you look, you know, if you summarize, basically you have a fermentation phase during which the cells will grow actively, okay? Then there is this particular phase that is called the deoxys shift, okay? During which the nutrients keep constant because the cells are basically stored, they rest the division cycle. And then they will resume their division cycle because they will actually, what they do is that during the first phase, the fermentation phase, they actually excrete ethanol, which is a carbon source. And because they prefer glucose over ethanol, they would excrete ethanol, keep going with glucose, but then when there is no longer any glucose in the medium, then they say, well, okay, maybe ethanol is not so bad, and they will start respiring ethanol, and this is what you observe during this third phase here, okay? So, and then, you know, once there is no longer any carbon source in the medium, then they go to this quiescence state, okay? So here the point that I want to make is, first, you know, the nutrient dynamics is very different when you deal with an unperturbed life cycle compared to an abrupt switch. And it's even more than that, because if you actually look at the ability of the cells to survive over a long period of time, okay, what you see is that the bona fide quiescent cells, they actually display a much longer, much better survival than the cells that have been abruptly switched to a no glucose medium. Okay, so if you're really interested in understanding, you know, how cells establish this quiescence program and what's the mechanism and so on, you'd rather go for an unperturbed life cycle because abrupt starvation just does not recapitulate, you know, the state of quiescence. Okay, so it's based on these considerations that we thought, you know, it would be interesting to be able to look into the true dynamics of entry into quiescence during an unperturbed life cycle. So for this, what we did was to use a microfluidic device. So this is actually very similar to the previous one, okay? But then what we did was to actually connect this microfluidic device to a population cell culture, okay, which you can see here. Pump some medium here. So basically the cells are growing there, okay? So they are depleting the medium from nutrients, okay? So the medium is pumped out of the flask and is used to actually feed the microfluidic device. Okay? And then, just for sake of maintaining the volume constant, we re-inject this medium into the flask. Okay? So this may sound, I mean, it's a very simple idea, right? The problem with this is that as the cells here enter a stationary phase, the cell number, the cell density becomes extremely high, like 10 to the 9 cells per mil or whatever. Okay? And this is a problem because this could actually clog the tubing and that kind of thing. So in order to prevent this, so my student had the idea of using one of these crazy you know, microfluidic device that allows efficient particle sorting. So partly it's a very standard thing. Don't ask me how it works. It's some sort of hydrodynamic effect. So you have this spiral thing, and over the, you know, the consecutive spirals, you end up enriching, you know, the internal part of the spiral is enriched with cells, and therefore we actually redirect the cells to the flask, and the cell-free part is actually used to actually feed the medium, to feed the microfluidic device. Okay, and we were able to show that, I mean, we were able to recapitulate previous results that make use of this device. And yet you can actually achieve a 100-fold decrease in cell concentration, which makes things much better in terms of device clogging and so on. So it's just a trick, but it was necessary to actually do this experiment. Okay. So with this device, so everything's going fine so far. I feel like maybe going too fast. So if there is any question, don't hesitate to harass me. Okay, so with this device, so the first thing we asked was, can we actually recapitulate the sequence of metabolic phases that I described before: fermentation, deoxic shift, respiration, and quiescence. Okay, and so this is one typical experiment. So here you see fermentation, and then the cell will stop completely, and then you see they will sort of resume growth, and the number of cells will actually increase, and then they will stop and forever. Okay. So indeed, we can definitely recapitulate. So you have you can see you have sort of looks like an exponential. I mean, it's not. Well, I mean, it's not far from an exponential, maybe. Then you have this plateau, and then the growth recovery here is like cells will be able to do roughly between 1 and 2 additional divisions before they actually enter a stationary phase. Okay, so we can recapitulate the sequence of metabolic phases, but then we wonder, can we actually get more insights about how metabolism is actually being changed during this life cycle? And for this, we thought maybe a pH sensor would be a good indicator, because pH, maintaining the pH is very critical for the cells, and pH and creating and maintain pH homeostasis requires a lot of energy, and therefore pH can be perceived as some sort of a very good proxy for the internal metabolic state of the cells. And so for this, we use fluorine, which is this engineered, GFP, modified GFP, which has an excitation spectrum that depends on the pH, and therefore we interrogate this GFP using 2 excitation wavelengths, and depending on the excitation spectra, depending on the pH, basically we can collect different amount of lights using different excitation wavelengths. Okay. So what we do here is that we basically follow the signal as a function of time, and we right away find something that was quite interesting to us, because we were able to see that not all the cells behave the same. Okay, and we could find two classes of cells that sort of diverge at the deoxic shift. So here you have one class of cells that somehow undergo this massive, huge drop of pH. I mean, huge. It's not huge, but it's pretty abrupt, and it's pretty consistent in all cells. So the cells undergo this drop in pH at the deoxic shift. Then it remains stable during the deoxic shift, and it actually increases a bit during respiration phase, which indicates that somehow, indeed, pH seems to be a good indicator for the metabolic status of the cells. Whenever the cells are growing, the pH is high. Whenever the cells are going to a low metabolic state, the pH goes down. Okay, and this is something that you notice further when the cells enter quiescence. You see that there is a further drop in the pH level. Okay, so that's one. Probably 90% of the cells actually would do this. Okay, but then there is this other 10% of the cells that actually undergoes this pH decrease right at the deoxic shift, similar manner as the other cells, but then they actually go further. Okay, and they reach a pH level which is the same as that of the blue cells, but much later. Okay, so we wonder what is going on in these cells, and I'm not going to show the details, but what we found was actually these cells are unable to switch to respiration. Okay, so they would stop right away at the deoxic shift. They would never enter the respiration phase, and this was actually triggered by the deoxic shift, because we were able to show that before, during the fermentative phase, they were actually respiration competent. Okay? So this is something that is triggered by the deoxic shift, and this is interesting because it's an interesting source of heterogeneity. So I'm not saying that all heterogeneity comes from this, but at least you can see that here this transition between different states is occurring at the deoxic shift is something that drives that promotes heterogeneity in the population. So this is one of the conclusions here. And then we wonder, yeah, just quickly, because if I want to have time for the rest, we know from previous studies that pH is a very important parameter for protein conformational state and the organization of the and also for the state of the cytoplasm as well. It has been shown that pH in using in vitro experiments or using experiments in which you can actually control the internal pH by permeabilizing the cells, that pH can induce this glassy light transition in the cytoplasm. So we were interested in looking whether the drop of pH that we've seen would actually induce cellular reorganization in the cytoplasm. And so for this, what we have done a number of markers, which I'm not going to detail. But what is interesting is that by looking at the signal, so the structure of the signal of this marker, what we have found is that indeed the entry into quiescence, so if you look up the blue population, so it's the respiration competent cells, okay, you see that as soon as the cells enter quiescence, you see that most of them undergo this, most of the cells undergo this transition to the formation of foci, okay, for all these markers, okay. And what is even more interesting is that if you look up the respiration deficient cells, so the red guys, okay, they also undergo this transition, but they do so much earlier. And so it seems that there is actually, if you look both populations, that there is a concomitance between the drop in the pH, which is represented with the dashed line, and the onset of aggregation of this protein marker. So this, with these other measurements, led us to formulate this idea that indeed during an unperturbed growth, the cells are actually experience this drop of pH that indeed is responsible for this transition of many cytosolic proteins. Okay, so another marker, and then I will switch to the next topic. So in order to assess the properties of the cytoplasm, people have used several different protein aggregate markers. So GLN1 GFP is one of them. This is known to be extremely mobile in cells that are not quiescent, but as you can see here, as the cells become quiescent or quiescent-like, just like this particular cell, the aggregate basically stops moving. Okay, so we have done this analysis in which we have looked into how fast actually the particle lose their mobility, you know, in response to the deprivation of medium. And what you can see is that all these respiration deficient cells, as you can see here, very quickly at the deoxic shift, the cells would lose their mobility, and this is actually concomitant with the drop in the pH. So the drop in the pH here is represented with the dashed line, okay, and the red points indicate when the cells lose the mobility of the aggregate. Okay. If you look now at the respiration competent cells, you see that this loss of mobility occurs much later, and it's also coordinated with the drop in the pH. Okay, so basically with this and the aggregation markers, so this led us to conclude that somehow the pH decrease that we observed and the glassy state transitions are sort of concomitant during this transition to quiescence. Okay, so that's the main conclusion, one of the main conclusions of this study. And yeah, so this is again all based on this idea that by using some combined approach, population scale and single cell approach, we can somehow use a single cell approach to quantitatively monitor readouts while having growth that resembles that of a population in a flask. Okay, so that's the main methodological insight. Okay, so now we'll just switch to the last part. So far so good, or do you have any question, or is it, no, not really. Okay. Yeah, sure.
So I'm just wondering about the biological function of this, and would it be some sort of bed hedging strategy, you think, to have some cells that are glassy. Sorry. You see these two fates, and I wonder if doing experiments where you make them escape from quiescence and put them back in, enrichment would they respond in the same way? Would they synchronize, for example?
Yeah, it's a good point. It's a great question. So I see two questions in there. One is, what is the function of this glassy state transition? As I said in the beginning, it is well known that quiescent cells are actually very resistant or very tolerant to stress. Okay, many different sorts of stress, and actually people don't know exactly why. Okay, and one simple hypothesis is that indeed, having this transition of the cytoplasm could actually confer, for some reason I have no idea why, but that these two things could be related. Okay, so this is one of the perspectives that we would like to pursue. Again, the increase in tolerance is like orders of magnitude, so compared to proliferative cells. Okay, so there is definitely something that is to be understood with respect to this stress resistance of the quiescent cells. Okay. However, so I don't have an answer, I have to say, because we haven't done the experiments. But the short term, the preliminary experiment that we did indicated that it's actually more complicated than this. So we did actually some stress response experiments with these cells. You expose the cells to hydrogen peroxide, for instance, an oxidative agent, at various timing during the quiescence. And we ask whether cells that have undergone this glassy state transition are more or less resistant than, okay? And the answer is that the glassy state transition does not do anything, because the cells already before the glassy state transition, they are already way more resistant than the proliferative cells. Okay, so we are a bit disappointed because we expected, if these two things are related, then it's a great story, right? But it's not the case.
Maybe they recover at different rates. That's what I was thinking. If you put them back in reach media, maybe there's.
Or maybe they recover faster. Yeah. So that, I don't know if we've looked into, we've looked into the resuming of the cell cycle upon nutrient replenishment, but I don't think I should have my student know. I have to say, I don't know the answer. If they, maybe they're listening actually. If you're there, you can just drop me a message. I'm sure they are. Anyway, I don't know the answer either. But this, okay, so now the second question about bet hedging. There is actually a paper that came out a bit earlier than ours, actually, from the Andrew Murray's lab. And he actually looked at abrupt starvation. And he actually claims in this paper that, so he sees the same sort of divergence of cell fate in the population. And he actually claims that these cells, which are unable to respire, so they are growing slower, they actually may be beneficial under some conditions when you actually switch the medium and so on. So he claims that there could be some underlying bet hedging mechanisms undergoing. So it's a current biology paper from last year.
So there's a message from Basile, said we did no difference in time of recovery.
You see? I was right. Okay, thanks Basile. Very handy. So I think time is good for me to switch to the next topic, which is the part dedicated to senescence. So we've talked about quiescence, which is a reversible proliferation arrest state. Now let's talk about senescence, which is by definition a terminal state for the cell. So if you look at textbook about cell cycles, and people will tell you, look, it's amazing how the level of refinement in terms of control of the cell division processes, all these checkpoints and so on. As you've seen, the ability of the cells to switch off the cell cycle, go back into and resume the cell cycle. All of this is very exquisitely organized. Okay, and so the question that we may ask is, okay, if cell cycle is a robust process, then why aren't cells immortal? And are cells immortal or not? Okay, so that's a long-standing question that we have in the lab, and actually this is also something that has been discovered long time ago that if you follow an individual mother cell over successive divisions, okay, you will see that after about 25 divisions, the cells will actually stop dividing, enter this senescent state, and eventually die. Okay, so this is referred to as replicative aging. Okay, so we are interested. It's an aging process in a way, which has to do with the ability of the cells to replicate itself. Okay. Okay, so this is something that has, as I said, been observed a long time ago, and the conventional way to actually do this kind of assessment is to use a microdissection assay, in which you use a microneedle to actually get rid of the daughter cells at each division, put the cells back in the incubator, wait another division time, which is about 19 minutes, and then do the dissection again and again. Okay, so that's a long-standing experiment. People are still using these kind of things, and we argue that there may be better tools. Okay, so a very important aspect of this replicative aging system is that the daughters of an aging mother actually recover a full replicative lifespan potential. Hence, you have some sort of interesting, germline effect with the daughter cell that ensures the survival of the species. And this can be viewed as an interesting asymmetrical model of division in which you have somatic cells that eventually enter into senescence, and a germline that self-renews the population. So one of the big questions that people have with this system is obviously what is the underlying mechanisms that drive this entry into cellular senescence, and whether this system is actually teaching us anything about aging in other species. Is there any conservation of the mechanism, and so on and so forth. I'm not going to go into the detail, especially in the second part, but I think it's always very important to have this in mind when you start developing a system, to ask, what is it that I'm working on, and what does it tell me and about the very system that I'm studying, and whether it would apply to something else, and what are the limitations of my model. Okay, so again, I won't go into the details, but one thing that I want to tell you is also from the methodological perspective, we always thought that these microdissection assays are very tedious, and they're also not much informative, because the only thing you get to know with this technique is at which age the cell is dead, but you don't know about the whole history of the cells over time. Okay, and so when I started to work on this more than 10 years ago, I thought, well, let's do a laser-based microdissection assay. So I developed this algorithm that actually keeps track of the cell controls over time, and that uses a laser to automatically dissect the progeny by killing the daughters. Okay, that's totally awful, right? It's very toxic to the cells because you end up, flashing, laser on the cells, and they actually live in symmetry, which they don't like at all. Okay, so I quit doing this, and I moved to something else after I realized, after I've seen this very famous publication, which is called the Mother Machine, that actually arose like in 2010, just 1 year after this. And in which people actually used some microfluidics, engineered cavities in order to trap individual cells and monitor their successive division over time. So this is an absolutely beautiful experiment, and also a pioneer experiment, which I can tell you that now in the field of bacteria, many people are actually using this kind of device just because it makes things much more standardized and much more simple to follow over time than looking at 2D colonies. So not much later, so different strategies have come up to actually trap individual yeast cells, so based on different geometries. And so in our case, what we developed is a system in which we have some sort of elongated cavity. So the cells will grow as a 2D colony, and eventually they will colonize this cavity. And once the cell is at the tip of the cavity, without going into the details, the cells may actually be stuck in there, and you can monitor its successive division over time. Okay. So the great thing about this, unlike microdissection, so is that we, well, like microdissection, we get about the same kind of survival curve as using the standard assay, so which is reinsuring. But the great thing about this is that we now have access to the whole dynamics of entry into senescence. Okay, so this can be represented using trajectories like that. Okay, so here you have individual cells, and what you already see is that by the end of their lifespan, okay, the cells tend to slow down their division rate. Okay. Based on this observation, what we thought is that basically all the trajectories of cells, despite the large variability among individuals, we could actually classify, a lifespan into two different states. One is the state during which the cells are able to divide normally, and the other one is a state, during which the cells actually slow down their division cycles. And you can see the statistics for the duration of each respective phase. And that led us to define what we call the senescence entry point, because it somehow illustrates the boundary between the two very different physiological states of the cells. So then the question became, okay, what is responsible then for this abrupt switch to a slow division mode that leads to senescence? What is the underlying mechanism? Okay. So we have looked into different hypotheses. So there were different papers that were putting forward different models. And one of the key models that was proposed a long time ago, so you can see it's 1997, was that the cells have this sort of Achilles heels, which is related to the instability of a genomic region in the genome, which is called the rDNA. Okay. So the rDNA locus is basically organized as a number of repeats, so roughly 150, 200 repeats. Okay. And that is just because a cell is basically producing ribosomes to make more ribosomes. Okay. And therefore, there is a huge burden related to the transcription of rDNA in order to produce ribosomal RNAs. Okay, so you have cells is constantly transcribing these regions, and therefore you have a huge number of repeats. Okay. Well, so what happens with this is that because there is a massive transcription, there is a risk that the replication fork would be colliding the transcription machinery, and so there is this replication fork barrier that actually ensures the stalling of the replication fork in order to let the transcription machinery go through. Okay, so this is great, but stalling a replication fork may not be very good for a cell, because this will actually lead to recombination events. And indeed, so it has been shown that sporadically the cells would actually undergo a recombination event that leads to the excision of single or multiple rDNA repeats. Okay, because these regions are repeated regions, recombinations is very easy. Okay, and therefore you may have the excision of these extrachromosomal rDNA circles. Okay, and furthermore, it has been shown that these ERCs are actually becoming more frequent as the cells get older, and not only they become more frequent, so they amplify, and they amplify because they have an autonomous replicative sequence, but they are also asymmetrically segregated between mothers and daughters. Okay. So this was presented as an interesting model to explain aging, because you could imagine that if the cells accumulate more and more ERCs, this would impact on the cell physiology over time. Okay. So this was one of the proposed models that was very that is still very successful. So we thought, can we actually monitor the evolution of the number of ERCs in single cells? Okay. So for this, what we did is that we took a strain in which all the repeats are actually tagged with LAC-O repeats, so that we can stain every single locus of the rDNA. Okay, so not only the chromosomal ones, but also eventually the ERC molecules. So we did this experiment in which we also used a nucleolar marker, because this rDNA locus is actually sitting within the nucleolus. So we use a separate nucleolar marker in order to quantify the green signal. Okay, so we get this trajectory. So, and if we collect several ones, what you can see is that it seems that there is indeed a relationship between the entry into senescence and this accumulation of this green signal, indicating that somehow the two things are related. Okay, so you can actually plot this as an average. Okay, this is what you have here. And interestingly, so the two things seems to be concomitant, but it's actually more precise. If you look at it more precisely, what you see is that the increase here in the signal actually precedes the onset of senescence, which is defined by the transition here. Okay, you can fit an exponential, and actually, if you subtract the offset here, what you observe. So there seems to be an increase in the number of RDNA copies, and if you subtract the baseline and you plot it on a semi-log scale, what you see here is that you have a very nice exponential over several orders of magnitude. Okay, and that's quite striking to have such a multi-orders of magnitude effect in a biological process, right? Or even though there is some noise here, obviously. But that indicates that indeed there seems to be an exponential accumulation of ERCs in the cells. Okay? And if you want, this is pretty much like what you get when you do a qPCR experiment, right? So here this is an in vivo qPCR in a way. So the cell is basically accumulating and mother is keeping for itself, but accumulating ERC molecules over time. So if you quantify the doubling time is about 1.5 generations, so it's a bit less rapid, or if you want, it's a bit less efficient than one division, probably because not all the ERCs are replicated within one division. But even more importantly, what you see is that if you extrapolate this line, okay, and if you ask when do I get the first extra RDNA copies, you see that it occurs at SEP minus 15 generations. Okay. So this for us is very important because this tells us that if the SEP is occurring here at division 0, that's the reference of timing, the ERC, the first ERC excision actually occurs much earlier than that. Okay? And therefore, it takes about 15 divisions for the cells to accumulate enough ERCs before it actually enters senescence. So there is a huge delay between the two processes. Okay? So, yeah, so, and there is, the cells has this extraordinary capacity to buffer the cell cycle defects during these 15 divisions. Okay. So, before I finish, so basically the whole model here is that the SEP is preceded by this ERC excision event, okay, that ultimately will drive a number of defects that I did not go into the details for the sake of time. But the current model is definitely the following, and the questions that we are now interested in is understanding whether this excision of the first ERC is a stochastic event, or whether itself is driven by other, age-related phenomena. So quickly, I understand that my time is about to be okay, but just a few words on this. Now, what we would like to do is to determine whether the first ERC event is actually a stochastic event that follows Poisson statistics and so on. So for this, we now have a strain that instead of having every single RDNA copy that is tagged, there is only one copy that has a transcriptional reporter. Okay? So we put this in the RDNA locus, and what we expect is that if the very RDNA repeat that has a tag is excised from the genome, then we would be able to observe an exponential accumulation of signal. Okay? But we expect this to occur only in a small fraction of the population because we have to be lucky enough that it's it's the tagged ERCs that is excised from the genome. So we expect that to see this kind of event in 1 over 100 cellular lifespan. So for this experiments, we have designed a different geometry of the trap so that we can monitor more cells. Okay, and there is one typical event of here that you can see has a preliminary result. You see that there is a massive accumulation of signal as a function of generation. If you plot it in log scale, you see that indeed there is this ERC accumulation. We can definitely time the excision, and therefore now the question is what is the distribution of these time between the birth of the cell and the excision of the cells. So this we have done actually quite preliminary, in a preliminary manner. So we have only 45 cells so far, and you see that, well, somehow it looks like an exponential. Okay? Okay? So basically, preliminary result here is that, yeah, it might be a stochastic event, so an age-independent event. So now we would like to do it in other mutants in which, for instance, ERC excisions is being modulated or segregation of ERC is impaired and that kind of things. Okay? But in order to do this, we would like to get a much better throughput because, as I said, so this is one event in 100 cellular lifespan, so we would need to analyze more and more data. And in order to do this, and I will finish with this, we have actually worked to make these experiments more automated and more high throughput. And as you've heard yesterday and also today, deep learning techniques have become very popular because they actually allow you to reach a much higher throughput in many different contexts. And therefore, with my student Theo, we have actually taken advantage of both microfluidics for image acquisition and deep learning to actually come up with a technique that allows us to really automate these experiments. So I'm just going to say a few words on this. So first, we have these traps that I just presented. So now we can monitor up to 3,000 cells times 16 different conditions. So we could be mutants, markers, whatever. So they are independent. Okay. Then Theo has built a simple microscope that allows us to actually monitor these traps in an efficient manner. So here you have a single field of view with a 20x objective. So this is just a zoom on one of the cavities. And we can image these cells. So then the question is how, if we can image all these single cells going through senescence, how can we actually extract the number of divisions, the lifespan, and so on and so forth. So basically the problem obviously is that, yeah, so my presentation is a bit screwed up, sorry for that. So here there will be a movie, but basically the idea is that the problem we have is that it takes about 1 to 5 minutes to count division during a lifespan, and that's extremely tedious. It's not very motivating for a PhD student and for anyone, I think. So, and people have done that for, we've been doing that for 10 years, but it's just that we end up having n equals 20, which is a pity, knowing that we can image 50,000 cells by now. Okay? So there was a big discrepancy between our ability to image and our ability to analyze. Okay? So the good thing is that counting divisions can be seen as an image classification problem. If you look at these cells here, you can attribute them a tag like small-bodied, small-bodied, large-bodied, large-bodied. And whenever the cells goes from large-bodied to small-bodied, it means that a division event has occurred. Okay? You can actually extend it and define other classes like this is a dead cell, this is a clogged environment, empty trap, whatever. Okay? So basically, we have defined a strategy that is based on the classification of individual traps, okay, in order to determine the state of the cells and count divisions. Okay. Well, so not going to go into the details of this. It's a bit of a mess, okay. But simply, this is just a simple CNN network that would extract features of the image that characterize the image. It's just a vector, okay, that characterizes the image. And instead of going from this image feature to the final classification, like small, large, empty, dead, whatever, we actually use an intermediate step, which is based on this so-called long short-term memory network, which is a kind of recurrent neural network. And the idea of this is to actually exploit the time dependencies between the images. Okay. So this is just one example of classification for 6 cells. So on the left you have the ground truth, and on the right you have the prediction by the algorithm. So it may be actually the other way around, I don't remember. And what you can see is that it seems to be very efficient, at least on these probably well-chosen cells, but no, it's not the case. If you look up more carefully, so we've done, so I think all these discussions about testing and checking, the outcome of these deep learning models and so on, I fully subscribe to this. I hope we have been careful enough. So this is, if you're interested, you can have a look. We have done a battery of tests, and actually what we have found out that is that actually using this combined architecture between a CNN network and an LSTM network provide very good accuracy, in the sense that, for instance, here, if you look at the correlations between what is predicted and the ground truth, you get pretty decent results, okay, which you don't get when you use only a CNN or different variations of CNN, okay. If you look at the survival curve, you can see that the agreement between the predicted and the ground truth is also very good. Division timings as well. So this is actually very, seems to be very promising to do this kind of analysis. And I will just finish by saying that on top of this, then you can use, the other algorithms that you're very aware of by now in order to extract, pixel information. So you can do semantic segmentations on top of that, and therefore combine, this automated replicative lifespan assays with the quantification of the signal using semantic segmentation. So this is my last step. I'm going to stop here. Just would like to thank the people. So the cell size control work was done by Cecilia Garmedia in the lab with the help of Nacho Molina in Strasbourg, with the theorist at the IGBMC, the chief theorist officer in the institute. The aging experiments were done by successive people in the lab, Stefan for the beginning, then Sandrine, and now Theo. The quiescence is a great project that was initiated by Basile and Theo. And the deep learning aspect was also done with Theo and a number of collaborators for the other topic. Thank you for your attention, and I'm happy to take questions. I'm sorry for taking so long.
No, it was great. Thank you, Gilles. Do we have questions in the audience? We should have questions.
Yes?
Do we know what is the mechanism that prevents the ERCs from being carried on from the mother cell to the...
Yeah. So there has been quite some work done there by a group in Switzerland, the Yves Barral group. And he has proposed this model that ERC tends to be tethered to the nuclear pore complexes. And the nuclear pore complexes, upon nuclear division, are not transmitted to the daughter cells. Okay, so that's basically how the model is formulated.
So some kind of affinity to the nuclear pore?
Yeah, so there is basically an intermediate between the nuclear pores and the ERCs. So what Yves Barral claims is that the saga complex actually itself is making the bridge between the nuclear pores and the ERCs. And that would be and so he has mutants in which he can perturb this inheritance of the ERCs and so on. But I have to say, this hasn't been really thoroughly validated by other groups. So as always, it would be nice to see whether other groups come up with the same kind of conclusion.
It seems like it's a complicated control to create, but in order to validate, it would be interesting to be able to create a mutant that does, transfer these ERCs to the daughter cell, and then validate that these daughter cells actually die pretty quickly.
That's right. So there is, to my knowledge, there is one key experiment that is close to what you suggest, which is people have actually used plasmids in which you can remove the centromere, but they still carry an autonomous replicating sequence, and therefore this synthetic plasmid would actually accumulate within the mother cells as well. And they have shown that by triggering, the excision of this centromere, they can actually induce premature senescence in the mother cells. So it's, in a way, it's a quite convincing argument that this accumulation of plasmids can be detrimental to the cells. And I forgot to mention that by the end of the lifespan, the cells actually have between 500 or typically 1,000 extra chromosomal RNA circles, which represent a huge amount of DNA, which is comparable to the genome size. Yeah. There was another question, no?
Just by curiosity, I was wondering, how did you actually test the germline-ness of the daughter cells?
Right. So it's a very old experiment. So what people have done is that so if you imagine this microdissection assay, so you let a mother cell age, okay, and at some point you take the daughter, and you actually do this similar experiment of replicative lifespan assay on a daughter of an old mother, okay, and you ask, does it recover a full replicative lifespan potential? And the answer is that if you take a daughter of a young mother, then it's fine. The daughter is fine. It has no detrimental effect on the daughter. But if you take the daughter of a very old mother, then the daughter is not doing very well. Okay, so that's one thing. So then you would say, okay, but then there is no rejuvenation anymore. Okay, but now if you take, instead of taking the daughter of a very old mother, you take the daughter of the daughter of the daughter of the daughter of a very old mother. Okay, so you go through the daughter lineage, and if you do this a number of times, then you end up ultimately having a recovery of the replicative lifespan. So the bottom line is there is indeed some sort of rejuvenation, so ability to recover a replicative potential through the daughter lineage. It may take a number of divisions, but ultimately cells will recover.
Thank you, Gilles. I think we can...